Hello everyone, this is a presentation of University of Florida course EEL 6825. My name is UQ and today's topic is cell boundary recognition using MATLAB. My project contains usually separates 4 parts. The first one is problem under study, the second existing approaches, and then performance evaluation of existing approaches, and the last, conclusion. For the first one, the problems. We all know that cell is a unit of tissues and organs in our body. A lot of experiments and analysis are being carried out in biological labs. Taking cells as the object of study means some steps in experiment procedures will include messy, boring, and repeated work. Taking cancer study, for example. In order to study some cell cycle or simply physical features in tens of thousands of cells, visual analysis of biopsy samples, histological slides, or medical images to note the difference from normal physiology seems to be a bad choice. Image processing demonstrates a huge potential in the field, so I wonder, can I use this image processing to deal with the cell works? So here comes image processing. Once I get an image, I need to deal with the noise in the picture, because there is some pixel noise resulting from some fluctuations ranging up and down with this true value. This fluctuation caused the noise. We can see in the picture there's a lot of white and dark dots surrounding the cells, and especially in the rest cycle. How to deal with this noise is a very important part, and the solutions I will talk about in the second section. Digital image format. Digital image format is a reflex of the amount of light one image can display during an exposure. In an image, the intensity information is stored in different lengths of storage space varying from the camera quality. Usually, we capture a 16 bytes image. And third problem, background subtraction. In a picture coming from a certain field of view, there are usually 2 main sources of error interfering image analysis. Cell debris, which can emit fluorescence signal, and illumination distributed in an uneven way. Background subtraction is the procedure dealing with these 2 errors. What we always do to subtract them is capturing the same field of view before placing the specimen. So we capture the features of debris and uneven illumination. Afterwards, place the specimen in this field and capture another image. We can subtract debris and uneven illumination features while combining these 2 pictures. In case of stationary state of debris and illumination distribution. In this condition, cell boundaries can be detected in a clearer way. We can see in the following pictures, the left top one is the original picture, and the other one is a picture after we do the background subtraction. We can see there's a huge difference between these 2 pictures. In the second picture, there's evenly illumination, and there's fewer debris of cell in this picture, which is much better for detecting than the former one. Okay, the second part, existing approaches. First, we need to capture the cell image. I get this picture from the electron microscope. The microscope, I use the eyepiece and objective lens are 10 multiply and 20 multiply is respectively. Exposure time is 300 milliseconds and image is 16 bytes. And then noise processing. Noise processing is very important in this image. I can use two kind of method to deal with the problem. One method is image cropping. What is image cropping? It is a quite basic and concise way to find out pixel noise. This method just need the user to choose a certain part of image. Make sure there's no target objects inside. Once capturing this noise features in Gaussian distribution, we can attain its mean and derivation. And thus, 95% of pixel noise will fall into this range. Choosing a proper threshold will be much easier. But it doesn't work in an evenly distributed illumination. How can we process the noise in an evenly distributed illumination picture? We'll use the following method. It is expectation maximization. This is a very important part in the course, and I want to emphasize this part. Expectation maximization is one method that can manage and estimate pixel noise in an image together with cell. Despite that, this method is involved in responsibility function and seems complicated. It is an efficient way to deal with pixel noise. And we're gonna use the following functions. This is a responsibility function. We can see the gamma equals pi multiply phi noise, and the bottom is pi multiply phi noise plus 1 minus pi multiply phi noise, phi signal. Sorry. And the parameters are defined as mu noise, mu signal, sigma noise square, sigma signal square. These are means and variance. Okay, after we deal with the noise, we can start to detect the object. Detecting the object, we use two steps. The first is to segmentation. It is to use auto method on segmentation to decide threshold. Once we decide the threshold, we can tell the boundary of the cell and the boundary of the background. It is very important to determine the right threshold. So auto method is very important. After we determine the object, we should need to detect the edge, and I use the operator to decide it. The operator is a Roberts operator. This can make the object's edge more obvious. Okay, here comes the most important part. This is a program, and this picture shown here is it is not the original picture. This is a picture after I determined the threshold. We can see the cells are highlighted in the middle, and the intensity decreases from the middle to the side. I can show you the original picture. Okay, this is the original picture. We can see there's one cell in the middle, and there's a nuclear in the center of the cell. The intensity is pretty high in the middle, and it decreases outside, but it's still higher than the background. Now I'm going to show how to determine the background, detecting the background and the boundary, cell boundary. Okay, this is a program, and in the middle it is code I have wrote for the project. These are several functions about maximum expectation, about DH filter, like auto method, and background subtraction. There's several functions here, and still I need two pictures to put cell pictures to put inside this program. One picture is I have just shown the black and white one, and the other is the nuclear of the cell. These two pictures, and right now I'm gonna run this program. At first it pumps out the question, how many positions here? The positions here means how many pair pictures I have provided. There's the one pair pictures here, one nuclear, one cell, so the answer is 1. And the second question, how many cells? There's just one cell. You can see here. Okay, this is the picture after we determine the threshold. That this is the section between the first step and second step. Okay, right now I need to set a point on the left top and another point on the right bottom. After I have decided the field to detect, it pumps out the result. We can see there's two pictures here. The left picture shows the boundaries of the nuclear, and the second the boundary of the cell. It works good, but maybe I need to make the line thinner. But in totally, this code works good. We can see in the picture, in the right picture, there is a cell on the left top, and maybe there is cell debris on the right bottom. But this program just regard them as a whole. Maybe I need to improve this. Alright, back to the PowerPoint slides. This is a picture after threshold. This is the result. To make a conclusion, I think this report works exactly what I need, but there's still some more place I need to improve. It is demonstrates the useful techniques for biochemical researchers to capture cell or other object outlines. But there's still some additional tasks can be done, such as enhancing the precision of the outlines by improving its threshold algorithm, or detecting various objects in a larger size range. Okay, right now this is the whole project I have done, and thank you for listening. Thank you.